Hello. In this video, I want to talk about synaptic activity, also known as synaptic transmission. You can think of this as the last part in neural activity. So part 1 is resting potential, part 2 would be graded potentials, part 3 is action potentials, and action potentials lead to synaptic activity. So if you recall, the whole point of an action potential is to cause the release of neurotransmitter. It seems like a lot of rigmarole to kind of go through to cause the release of neurotransmitter. And I agree it is, but it works, right? So that electrical activity of the action potential causes the release of neurotransmitter. Neurotransmitters will then open receptors on another neuron. Don't forget that receptors are typically chemically gated ion channels. So neurotransmitters are released at an area called a synapse. A synapse is just a fancy name for a meeting place between the end of a neuron and another cell. So this is where a neuron will communicate with another cell at a synapse. An example of a synapse we've already studied in our course is the neuromuscular junction. So the neuromuscular junction is a synapse between a neuron and a skeletal muscle cell. Now, during neural activity, we've been concentrating on neuron-to-neuron communication as our example, so we will continue with neuron-to-neuron communication here. So in this case, the cell that's being communicated with is going to be another neuron. So you can think of a synapse as having 3 parts to it. Part 1 is the presynaptic neuron's axon terminal. This axon terminal contains the synaptic vesicles that are going to release the neurotransmitter. The second part is going to be the synaptic cleft, which is the space between the 2 neurons. And the third part is the postsynaptic neuron's dendrites, which has receptors for the neurotransmitter. So in the picture here, we can see the axon terminal of our presynaptic neuron, and you can see the bubble-like structures in the axon terminal that contain the neurotransmitter. We see a space called the synaptic cleft, and then in green, we see our postsynaptic neuron. So this is likely going to be the dendrites or possibly the cell body of our postsynaptic neuron. And notice that it contains receptors for the neurotransmitter. So the events of synaptic activity are rather simple. And if you recall the use of a neurotransmitter at the neuromuscular junction, you're pretty much going to know almost all of the events that are occurring here during synaptic activity. Okay? So let's start off by looking at how we connect the action potential to the release of neurotransmitter. Now don't forget the action potential had different phases to it, a depolarization phase and a repolarization phase as its 2 major parts. When the depolarization phase of an action potential reaches the axon terminals, that change in voltage that's accompanied by that depolarization is going to open voltage-gated calcium channels. Right? So what opens a voltage-gated calcium channel is a change in voltage. What goes through a voltage-gated calcium channel is calcium. So that depolarization will open the voltage-gated calcium channels, and this will allow calcium ions to follow their concentration gradient. Don't forget, calcium ions are concentrated extracellularly. So if we open up a calcium channel, the calcium ions will move from their area of high concentration outside the neuron to their area of low concentration inside the neuron. So the calcium ions are just going to diffuse into the neuron through the channel. Once the calcium ions are inside the neuron, they act as an intracellular messenger. So calcium ions are a very common intracellular messenger. We saw this in muscle physiology when the calcium would bind to troponin. So here calcium is going to trigger the exocytosis of the neurotransmitter. So calcium causes the synaptic vesicles containing the neurotransmitter to move to the cell membrane and then release their contents via exocytosis to the outside of the axon terminal. Basically, the neurotransmitters are released into the synaptic cleft. Now the neurotransmitters will diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. The neurotransmitters, when they bind to their receptor, they are going to open that receptor. Typically, the neurotransmitter receptors are chemically gated ion channels. So the neurotransmitter acts as the chemical that opens the gate. And now ions will either rush into or out of the postsynaptic neuron, causing a change in voltage in the postsynaptic neuron. And now what we've done is we've started a graded potential in the postsynaptic neuron. And that graded potential may or may not lead to an actual potential in the postsynaptic neuron. I wanted to mention that after synaptic activity is completed, the neurotransmitter is either broken down by enzymes in the synaptic cleft, or it can be reabsorbed through a process that we call reuptake by the presynaptic neuron, so that the presynaptic neuron can reuse the neurotransmitter. Or sometimes the neurotransmitter just diffuses away. Let's take a look at this process with a pictorial format. Okay? So this is going to be our presynaptic neuron. So this is the neuron that releases the neurotransmitter. Here's our synaptic cleft, and this is our postsynaptic neuron, the one that receives the neurotransmitter. Okay? So this red arrow is denoting the depolarization phase of the action potential reaching the end of the axon. Now that depolarization is what's going to cause the change in voltage that will open our voltage-gated calcium ion channel. Calcium ions then move from outside the neuron to in, following their concentration gradient. Once the calcium ions are inside, they cause the release of the neurotransmitter. So these little orange dots are the neurotransmitter being released into the synaptic cleft. The neurotransmitter then travels across the cleft and binds to its receptor. So these blue things are the receptors for the neurotransmitter. The receptors for the neurotransmitter are typically chemically gated ion channels. So the neurotransmitter acts as the chemical that opens the gate. Then the ions will flow into or out of the neuron depending upon the type of channel and cause a change in voltage in our postsynaptic neuron. So I've mentioned that once the neurotransmitter binds to the receptor, we can have ions flow into or out of the neuron, and it sounds pretty contradictory almost. So I want to talk a little bit about that. So the effect of a neurotransmitter on the postsynaptic neuron depends on the receptor for the neurotransmitter. So the receptor type is what determines the effect that the neurotransmitter will have on that postsynaptic neuron. The receptor will determine if the neuron experiences an IPSP or an EPSP. If the neurotransmitter receptor opens sodium channels, depolarization will occur, and the postsynaptic neuron experiences an EPSP. If the neurotransmitter receptor opens potassium channels or chloride ion channels, hyperpolarization will occur, and this means an IPSP will occur. So you can see how the receptor is really important for determining the effect on the postsynaptic neuron. Let's take a look at a specific example. So we're going to take a look at cholinergic receptors. So cholinergic receptors are receptors capable of binding to acetylcholine. So acetylcholine is one of the many neurotransmitters used in our body. We've talked about acetylcholine before in our muscle physiology section. So cholinergic is just the adjective form of acetylcholine. So it turns out there are two types of cholinergic receptors. They're called nicotinic receptors and muscarinic receptors. And their names are really funny, but actually their names come from the fact that these receptors can bind to other things besides acetylcholine. For example, nicotinic receptors bind to both acetylcholine, and they can also bind to nicotine. Anyways, that's not my point here. My point is this. The nicotinic receptors tend to be chemically gated sodium channels, which means when acetylcholine binds to a nicotinic receptor, sodium is going to rush into the neuron, causing an excitatory postsynaptic potential, because when sodium rushes in, it causes a depolarization. The muscarinic receptors, though, are very different. The muscarinic receptors, when they bind to acetylcholine, they don't usually lead to the opening of sodium channels. Instead, they lead to the opening of chloride ion or potassium ion channels, both of which will lead to hyperpolarization of the neuron and an IPSP. Let's look at these in pictorial formats, right? So this is how a nicotinic receptor might work. So here is the nicotinic receptor. Here we can see that the acetylcholine is now bound to the receptor, and the acetylcholine acts as the chemical that opens the gate, and now sodium ions are diffusing into the neuron, and that will cause depolarization of the neuron, leading to an EPSP. This is pretty typical for how a neurotransmitter might work. Now here's the muscarinic receptor, and this is sort of a little bit of a different situation. Here, the receptor is not a chemically gated ion channel, right? And most of the time, the neurotransmitter receptors are chemically gated ion channels, but not all the time. So the gray, boxy looking like thing is representing our muscarinic receptor. And when acetylcholine binds to the muscarinic receptor, it activates something else inside the cell. And this something else opens a chloride ion channel. So we are still opening an ion channel here. So that remains the same. It's just that the ion channel itself is not binding to the neurotransmitter. But anyways, the important thing I wanted to emphasize here is that we're opening a chloride ion channel, which means negative ions are going to rush into our neuron. And if negative ions rush into the neuron, we get hyperpolarization, not depolarization. So we're going to get an IPSP. So acetylcholine is causing a depolarization here, and acetylcholine is causing a hyperpolarization here. The same neurotransmitter is causing two completely different effects. And this is important to understand because it's not the neurotransmitter that determines the effect on the neuron. It's the receptor. What's different between these two pictures is the receptor, right? So the receptor type is really important for determining how the neuron will respond. Now there are lots of other neurotransmitters besides acetylcholine. Maybe you've heard of serotonin. Serotonin is thought to play a role with things like our mood and our attention. Low levels of serotonin are associated with depression. People that take antidepressants are actually taking a drug that inhibits the reuptake of serotonin into presynaptic neurons, causing serotonin to hang out in the synaptic cleft for longer periods of time, enhancing the effects of serotonin in those individuals. Dopamine is another neurotransmitter you may have heard of. This one is really important for signaling in our brain for motor control, so when we want to send out messages for moving our muscles. But it's also really important in the brain for our pleasure signaling. Opioids play a role with dopamine. Epinephrine and norepinephrine are also neurotransmitters. They're very important for our fight or flight response, and you might know them better as adrenaline when we use them as a hormone. GABA is possibly another neurotransmitter you might have heard of. GABA is said to be a major inhibitory neurotransmitter in the brain because it tends to cause IPSPs in neurons. Now, before we leave the idea of synaptic activity entirely, I want to mention that so far we've been studying a type of synapse called a chemical synapse. And it's called a chemical synapse because we release a chemical in the form of a neurotransmitter into the synaptic cleft. But electrical synapse do exist, and electrical synapse is found where the axon terminal of one neuron actually touches the dendrites of the postsynaptic neuron. And there's no release of neurotransmitter. Instead, electrical activity travels from the presynaptic neuron to the postsynaptic neuron. So we just have an action potential that travels from one neuron to the next. This is not common, but it does occur, so I did want to mention it. All right, let's see who we have here. We have Kurt Cobain, of course, the lead singer of Nirvana. Stephen King, famous author. Cindy McCain, who is the wife of the recently passed, or maybe not so recently, depending on when you're watching this video, Senator John McCain. And then we have Prince. And then the one at the end is probably the hardest. That is Thomas Jefferson. So what do these five individuals have in common? I mean, obviously they're all people, but they have something else in common. And they're not all dead, right? Stephen King's still alive. Cindy McCain, still alive. Opioids. All of those individuals had an opioid addiction, right? So I want to talk a little bit about opioids. Opioids are chemicals that bind to receptors called opioid receptors. And what's interesting is that we all have opioid receptors at our axon terminals. Okay? So in the picture here, we see a picture that I've showed you before to talk about synaptic activity. But what I added in was this little orange barrel, which is representing our opioid receptor. Okay? So our presynaptic neuron has opioid receptors at the axon terminal. These are receptors that cause potassium channels to open. So when the opioids bind to that receptor, potassium channels open, and then this allows potassium ions to leave the axon terminal, and that causes the axon terminal to hyperpolarize. Now, the hyperpolarization of the axon terminal prevents depolarization of the axon terminal, right? So remember, what's supposed to happen at our axon terminal is the action potential arrives, that depolarization phase of the action potential arrives, opening voltage-gated calcium channels, and then the calcium triggers the release of neurotransmitter. But what the opioids do is they cause hyperpolarization of the axon terminal, preventing the calcium from entering, which prevents the release of neurotransmitter. And this completely stops the signaling pathway. This is great when you have horrible pain, because the opioids will completely stop these pain signals from occurring. Unfortunately, opioids have other effects too. So not shown in this picture is the fact that opioids actually stimulate pleasure pathways in the brain, and that's what leads to addiction. So opioids can be very dangerous because they are very highly addicting. So take a minute to check your learning, pause the video here and answer these questions, and then play the video to see if you got them right.